Okay, now we're going to talk about cell respiration. So cell respiration, remember, occurs in the mitochondria. That's the powerhouse of the cell where food energy, primarily carbohydrates or glucose, are converted into energy currency, this molecule ATP, that our cell uses directly to power all cell activities. Okay, so overview this process right now that occurs first in the cytoplasm and then in the mitochondria. So let's first talk about when we talk about cell respiration. When you think about respiration, you think about breathing. Breathing in and out, that's respiring. But cell respiration is really what's going on at the cellular level. So the first thing when we eat food, let's say you eat some peanuts, okay, you digest them, right? So peanuts have a lot of proteins, and those proteins are broken down into amino acids. Peanuts also have a lot of fat. Fats are broken down, they're triglycerides, so they're broken down into glycerols and fatty acids. And there's some carbohydrates in peanuts too. So those carbohydrates are broken down into simple sugars, what we sometimes call monosaccharides. Okay. Primarily, we're going to talk right now about how sugars are used for food, since that's the main energy source we use. We use carbohydrates, which break down into glucose, and glucose is kind of the main energy food that we use to make ATP. But then at the end we'll talk about how proteins and fatty acids and glycerols are also used in a similar process or in parts of the cell process to get energy to make ATP. Okay. So step one, when we start with a glucose molecule, okay, the first thing we're going to do is called glycolysis. Glyco just means sugar, and lysis, as in lysosome or hydrolysis, means to break up, to split apart. So glycolysis is basically splitting a 6-carbon glucose into 2 3-carbon molecules called pyruvate. And this occurs in the cytoplasm, so we're not yet into the mitochondria. This is just enzymes out in the cytoplasm chopping up glucose in half. And when we do that, we release some energy. We get energy out, and we can use that energy to make some things. We can make 2 ATP, which again is what we want to make here to power all cell activities. But we're also going to strip off some electrons and some hydrogen and make this molecule NAD. And that's important because that's going to come in our last step of this process where we harvest that energy. So we've made 2 ATP, we split glucose into 2 3-carbon units, and we're still in the cytoplasm. Okay, not much energy that we've extracted out of glucose, but it's a good first start. And this is called, again, glycolysis. Step 1, splitting sugar in half. Okay. Step 2, now we're going to move these 3-carbon molecules into the mitochondria, and we're essentially going to chop them up into carbon dioxide. So how many carbon dioxides can you make from a 3-carbon pyruvate? Well, you can make 3. So if we have 2 of them, that's going to total 6 carbon dioxide. So cell respiration, we take a sugar and we chop it up into 6 carbon dioxides. And we do that, we're going to also generate 2 more ATP. So we got 2 from, again, chopping up and releasing energy by breaking those bonds. And we got 2 ATP in glycolysis. So far we have 4 ATP produced, okay? So that's pretty good, but there's a lot more energy. There's a lot more hydrogen and their electrons in there that we have not yet extracted energy from. So we'll talk about what happens there. So a little more detail about the Krebs cycle. So here's our pyruvate, the 3-carbon molecule. It goes into the mitochondria. So here's the mitochondria. And it gets chopped up into a 2-carbon molecule, release of carbon dioxide right there. And then it gets fed into this carbon cycle where it gets chopped up into, you can see the remaining 2 carbon dioxides. So there's 2 of these. So there's 1 carbon dioxide there. There's 2 there. There's our 3 carbon dioxides per each 3-carbon pyruvate, but we're also stripping off the hydrogens and their electrons. And again, we're using them to reduce these high energy carriers. These are basically just B vitamins, but they're going to actually sort of carry some of the electrons and their energy and with the hydrogens and the electrons to the last step, and that's the electron transport chain. And we'll talk about that in a second. So we have glycolysis, we've split glucose into 2 pyruvates, and we've got 2 ATP. We then have fed the 2 pyruvates into the Krebs cycle. We've got 2 more ATP, but again, we've got all these hydrogens that we've extracted with their high energy electrons. And now we're going to pass those to this series of proteins on the inner membrane. Remember the mitochondria when we studied the cell, has this inner folded membrane right here, okay? And it's full of these enzymes, and what these enzymes do is they basically take these electrons and they extract energy from them and use that to make a lot of ATP, and it's called electron transport chain. Okay, so that's the last step of cell respiration. Okay, so a quick analogy for you. If you've ever seen a picture of the Hindenburg, right? The Hindenburg was a big blimp filled with hydrogen, and it kind of scraped the ground, and there was a spark or some heat, and it blew up into a mass explosion. So we know that hydrogen has a lot of energy in its electrons, but in those covalent bonds, right? What you need is you need to combine it with oxygen, and you can release that energy, and that was the explosion of the Hindenburg, right? And that produced a waste product called water. So hydrogen and oxygen combined released massive amounts of energy and produced water, H2O, okay? Well, we do that in our cells, but we don't want massive explosions. We'd blow up, right? So we actually slowly extract that. So what happens, these proteins in this inner membrane that I just talked about slowly kind of grab these electrons and pass them down slowly. So you can think about a more controlled reaction where we're going to slowly extract that energy. And that's what this electron transport chain does. Each kind of step as it gets passed down generates some ATP, right? And when we're done, we get a lot of ATP produced in this process. And when we're done, we have the electrons and the hydrogen that gets combined with oxygen. So this is where we need oxygen in cell respiration for this last step to make the molecule water. So we're essentially doing what the Hindenburg did, but that was an uncontrollable explosion. Our cells are doing it in a very slow and controlled way and using it to make a lot of this important molecule ATP. So that's the process of cell respiration. We have first glycolysis, we have the Krebs cycle, and then this last step of the electron transport chain. Now we're going to talk about what happens when we don't have oxygen in cells. So if it's your brain and we denied oxygen, the cells die very quickly and you're dead. But we have other cells that have adaptations for surviving without oxygen. So if we have oxygen, we're going to go into the Krebs cycle and electron transport chain, and we call that aerobic respiration, A-E-R, oops, my pen's not working, A-E-R-O-B-I-C. If we don't have oxygen, it's going to be anaerobic, A-N-A-E-R-R-O-B-I-C. Did I do that right? Anyway, anaerobic, A-N-A-E-R-O. There should be an O in there. Sorry about my terrible writing. Anyway, anaerobic means without oxygen, okay? So let's talk about two of the processes that we're going to see in the biology lab, or that we'll talk about. First off, we take yeast and deny them oxygen. They'll go through a process called fermentation. So fermentation is basically cell respiration in an anaerobic environment. We deny them oxygen, and instead of producing carbon dioxide and water, they produce something called ethyl alcohol and carbon dioxide, which basically is what beer is. And if we let the CO2 vent out, we make wine. So let's look at how that occurs. So we have glucose, right? And we have this first step, glycolysis, exactly the same. We make 2 ATP, and we make 2 pyruvate. Okay, so glycolysis, it exists. It's an anaerobic process. Whether or not there's oxygen, glycolysis is always going to occur. Okay, great. Then we're going to take that 3-carbon molecule. We're going to chop off 1 carbon dioxide for each of those 2 pyruvates for a total of 2 carbon dioxides. And we're going to have a 2-carbon molecule that we then are going to convert into ethyl alcohol, a 2-carbon molecule. There you see the 2 carbons. Okay. Now this is a high energy molecule. We can actually take this, and we do take this and put it into our cars and burn it for fuel. So this is not a low energy waste product. It's also a toxic waste product. So yeast don't want to produce a high energy toxic waste product, but if they're in an environment with no oxygen, it's better than death. So that's what they do. They sacrifice a less efficient process producing a high energy toxic waste product, but enables them to survive. And they produce some carbon dioxide and they produce ethyl alcohol. And again, we use that to make beer, wine, and all fermented and distilled beverages, alcoholic beverages. Okay. Now in our bodies, if we did that, if we produced alcohol, we'd get drunk if our cells were doing something anaerobically. And that wouldn't be very good if you were like, running from a bear and you were getting drunk and you were stumbling and falling and, and getting dizzy. So our body does something else. We'll take glucose And exactly the same thing, glycolysis, we get our 2 ATP right there, and we get our 2 pyruvate. Again, glycolysis is the first step in all processes, aerobic or anaerobic. But then we'll convert that 3-carbon pyruvate into a 3-carbon molecule called lactate, and it's sometimes called lactic acid. Now, lactic acid is what you feel when you're running and your legs start to burn, or when you're in the weight room and you're overexerting your muscles and they can't get enough oxygen and they start to burn. So it's that acid. Acids we know burn and irritate and are damaging to proteins and tissue. So it's that burning sensation. It's actually that acid that's produced in our cells. But again, if you're running from a bear, it's better to get a burning sensation in your muscles than to have muscle fatigue where your muscles don't work at all and you are eaten by the bear. So we sacrifice, again, efficiency, and we produce a toxic, painful substance, lactic acid, but it allows us to still produce some energy for our muscles to flee bears. Now, this is something that our muscle cells can do. Our brain cells cannot, so we deny oxygen to our brain, we're dead. But it's another adaptation that allows for making ATP without oxygen, and we call that an anaerobic process. Okay, and then lastly, I talked about how we also can use fats and proteins, not just glucose, to make energy. And quickly here, it's amino acids are these 3-carbon chains, and we can take them and we can chop off the amino group, and then we can feed them in here at the pyruvate section, or we can chop off a carbon and feed them in at this section. Fats are composed of glycerol and fatty acids. The glycerol is a 3-carbon unit. We can feed it into here. It's very similar to an intermediate product of glycolysis, and that gets broken down. Fatty acids can get chopped up into 2-carbon molecules and fed in here, and they're long chains, so there's a lot of energy in all those carbon-carbon bonds. So there's a lot of energy in these molecules. They're not as oxidized as carbohydrates, so they don't burn as cleanly, but we can use fats and proteins, not just glucose for energy. It's just a matter of feeding them into different steps of this process. But when we're talking about carbohydrates, our main source of energy, we're talking about glycolysis, we're talking about the Krebs cycle, and then the last step is the electron transport chain. That's the 3-step process for cell respiration in our cells within the mitochondria. And that's it.